Okay, well now that we've reached the 4 o'clock hour, we're going to go ahead and get started. I'd like to thank everyone for joining us today. This is the National Ocean Sciences Bowl Professional Development Webinar Series. If you don't know anything about the NOSB, it is an academic competition for high school students where they get tested in their knowledge of all things related to ocean sciences and the ocean. And we offered this webinar series specifically for our coaches to help them prepare for the competitions as well as learn new things to take back to teach their students who will be competing, and also to be able to infuse this information into any of the classes that they may teach. But we love to have all educators involved and watching the webinar, so whether you're an NOSB coach or not, I hope you'll enjoy the webinar today. Every year, the NOSB has a competition theme. The theme for this year is the science of oil in the ocean, so therefore our presenters the past few weeks have been presenting on things related to oil in the ocean, and today we're sort of focusing on the ecosystem impacts. The webinar schedule, this is basically an hour-long presentation by our guest expert, and then we'll have a series of 30 minutes for question and answers, and so you'll see there is a chat box on the webinar screen. So during the presentation or after, feel free at any time to type in your questions. After the hour presentation is up, I will read them aloud to our presenter. We currently have everyone muted just so that we can hear the presenter and there's no odd noises in the background. While you're watching the presentation, you'll notice that you see the screen with the presentation as well as participants and the chat box. You can close or minimize the participants and chat box and use the two little arrows that look like they're going in opposite directions from each other to maximize the presentation to the full screen, if you so wish. Also, please feel free to provide feedback on either what you enjoyed in the webinar or send us questions at our email inbox, which is nosb@oceanleadership.org. We love to hear what you think of these presentations. Your comments certainly help us retain funding for this series, and they also help us shape what we'll do in future webinar series. Today, I would like to introduce our guest expert. It is Dr. Joseph Montoya, and he is from Georgia Institute of Technology. And today, he is going to be presenting on the impacts of oil and gas on offshore ecosystems. So with that, Joe, I'm going to turn it over to you.
Okay, thank you, Melissa. Delighted to have this opportunity to talk to people. And what I'd like to do is share with you some of the work that's been carried out and is still ongoing in my lab, which focuses on the ways in which oil and gas can affect offshore ecosystems, primarily up in the water column, but we do some work as well down on the bottom. And all of this work, although it's fundamental science, does have implications for fisheries and things that humans are actually interested in. I'd like to start off by acknowledging our funding sources. NSF supported some of the early work I'll be showing you, and much of the work I'll show you, that I'll share with you, has been carried out by the EcoGig Consortium. I'm a member of that consortium. It's funded by the Gulf of Mexico Research Initiative and is a concerted effort to try to understand how oil and gas released into the natural environment affect ecosystems. So my plan for today is to start by telling you a little bit about how oceanic food webs are structured, who some of the important players are, and then I'll share with you the way that we study these food webs, give you a very quick tour of some of our cruises and some of our results, and I'll wrap up by spending a few minutes talking about both the ecosystem impact and some of the broader societal impacts and concerns of oil and gas in the open ocean. That image down at the bottom is a picture taken in late September, mid-September actually, 2010. We were just about 1.5 miles from the Deepwater Horizon wellhead, and what you see in the background are some of the drill ships that were drilling the relief wells that actually led to sealing of the wellhead in the end. This is an image that some of you may have seen. It was a schematic put together by the Coast Guard and NOAA to try to capture some of our thinking, some of our understanding early on of the various impacts of oil and gas on open ocean systems. Now, our bias, I think, has always been that much of the impact of oil and gas will be up at the surface and at the coastlines. And in this image, you can certainly see, and I'm going to use my cursor to point things out. You can certainly see that stuff is happening down at the seafloor at the wellhead. There is a plume of oil ascending to the sea surface, creating all sorts of problems. We can see big oil slicks, we can see burning stuff, and we can potentially see oil running ashore where humans really care about it. We can also see that some of the oil may move offshore, and some of it may move out at depth. This was initially unexpected, and in fact there was substantial debate initially over whether or not oil could persist at depth in substantial quantities. This figure also captures a couple of things that I'm particularly interested in. So there's a whole lot of biology and chemistry captured inside those two red boxes I've just outlined. Perhaps animals, copepods in this case, then consume some of the oil or oil-derived carbon and excrete it, metabolize it, do something with it. And there may well be other biological and chemically driven pathways that will lead to oil being captured and transformed into sinking particles, some of which at least may make it all the way down to the sea floor. This was a very early attempt to capture those ideas, and in fact we know now that all of these processes are important, and in fact that a fair bit of oil ended up in these midwater ecosystems and down on the sea floor. Now, one thing that's not shown here is any sense or any concept of a food web, and yet we know that feeding processes, such as this consumed by copepods business, are very important in both the ecosystem response to an oil spill and in the overall impact of that oil spill. One of the reasons for that is that it's really hard to study these food web impacts. I'm an oceanographer. I'm a biological oceanographer by training, but I work at the interface between biology and geochemistry. And here's just a few photos to give you a sense of how it is we work out at sea. The cruise that we carried out right after the oil spill was on the Oceanus, a vessel out of Woods Hole Oceanographic. And it's an intermediate-sized vessel, and we filled it with colleagues and equipment. So the other three images show you just three images of colleagues and gear in the main lab. It was a crowded ship. We, as I said, packed it full of people, and it gave us the opportunity to really study the immediate impacts of this massive release of oil and gas in the Gulf of Mexico. You're going to hear about these sampling devices repeatedly through this talk. The top center image shows what is that? That's the recovery. I had to peek to make sure it's a recovery of one of our standard sampling packages. This is a CTD rosette. CTD stands for conductivity, temperature, depth. That refers to an electronics package down at the bottom of the frame, which in real time gives us information on the physical and many of the chemical properties of the water column. In addition to that sensor package, there's an array of sampling bottles. This image at the left shows it in side view. Typically, we have 24 10-liter bottles, and we can trigger them, close them, at selected depths in the water column to give us samples of interesting features. Now, that lets us collect water for chemical work, for experimental work. It doesn't really let us sample the food web itself. To do that, we have to use nets of various sorts. And on the right-hand side, I have an image of a particular kind of net, and I'll show you more of this in a bit, but it's a net that allows us to capture bigger organisms at different depths in the water column, and to do so in a quantitative way so that we understand both what the kinds of organisms in the water column is and what their abundance and concentration are. Here's a brief timeline of the cruises I'll be showing you. So the Deepwater Horizon spill began in April 2010. The mass of the spill really ended by mid-July 2010. The wellhead was declared sealed in early September 2010. We were out there in August, September 2010, immediately after the bulk of the oil had been released. Again, we were on the Oceanus, and we sampled in a region very close to the wellhead as well as farther afield. We returned in 2011, different ship with similar capabilities, and surveyed not just the immediate vicinity in the Northern Gulf. In 2012, we returned again, surveyed in the vicinity of the wellhead and again further afield. We've gone back in 2013 and 2014, and just for context, here are a couple of the cruises we carried out. In 2013, we sampled the water column of our CTD rosette. We used the mock nest to sample zooplankton. We use a multi-core to collect sediment samples. And we have a sediment trap that lets us tie what's happening in the water column to what's down on the sea floor. On all of our cruises, we measure microbial activity. We carry out experiments to begin to understand what's happening with the ecosystem when we add oil. And we measure a variety of microbial and plankton rates. Now, I told you that there's a lot of biological activity captured inside these two red boxes, but you don't see anything resembling a food web. And one of the reasons for that is food webs are complicated. This is a classic figure that shows an attempt to capture the feeding relationships of North Atlantic cod. And don't worry about any of the labels, just take my word for it that this is an attempt to really crystallize what it is cod feed on and how those organisms are related one to another. It's a mess. There are multiple connections, multiple species, and it's very, very hard to really tease apart how the pathways are interconnected and which the dominant pathways are. We inevitably have to simplify those food webs. So here's a couple of traditional ways to look at pelagic, that is water column food webs. The upper part of this figure shows what we typically find in open ocean systems. On the left-hand side, we have primary producers. On the right-hand side, we have the big, economically important fish that people typically care about. And connecting them, we have a variety of zooplankton, starting with protists and going up to various sorts of crustaceans and other mid-sized zooplankton, and finally leading us to fish. On the continental shelves, there are typically fewer steps connecting the primary producers to the big fish that we care about, but there's also a benthic component, a seafloor component, that can also be important in channeling energy and material from the primary producers up to those big economically important fish. Very few people who aren't scientists or interested in science know or care about all of these links. In fact, what people typically care about are things like this. This was one of the seamen on the Oceanus, and he captured those two mahi in about 5 minutes while we were out there, actually fairly close to the wellhead. But remember, those are the fish that people typically care about, but we need to have the movement of organic matter from the primary producers up to those big charismatic fish in order for those big charismatic fish to actually grow and produce biomass that we can hope to exploit. So my lab and our colleagues are studying the ways in which oil and gas released into the environment affect the primary producers and the parts of the food web that lead up to those big fish that people care about. All right, let's start by looking at some of the impacts of oil and gas on the bottom end, on the primary producers and the small organisms that really start that transfer of the impact from oil released into the environment up towards the bigger organisms. Here are a couple of students from my lab. We routinely go to sea and we filter tremendous, large quantities of seawater, tens to sometimes hundreds of liters, in order to get a feel, an understanding of how oil and gas are contributing and affecting the primary producers. When we do that, we are collecting water from that CTD rosette, and we target our sampling to give us samples of interesting features. The figure on the left is a screen capture from one of our deployments of the CTD rosette. This was done in the Northern Gulf, a few tens of kilometers away from the Deepwater Horizon wellhead, a few months after the wellhead was sealed. The part of this figure I'd like you to focus on is that red trace at the right-hand side of the panel. That represents the beam transmittance. So we have a device on the CTD rosette that measures the clarity of the water. Those leftward going peaks that you can see here at about 100 meters, 200, 300, close to 400 meters are portions of the water column that are very turbid. And in fact, we used a submersible camera system to confirm that these turbidity features were associated with very high particle loads. We then collected water using the sampling bottles on the CTD rosette and characterized it biologically and chemically. Now, the kinds of organisms we capture in the sampling bottles include all manner of phytoplankton, as well as lots of inorganic particles. The phytoplankton are the primary producers, and particularly up near the surface, they constitute a very diverse, very abundant, very dynamic community with a whole range of different kinds of organisms. So this is just a generic cartoon showing some of the abundant kinds of phytoplankton. And in the Northern Gulf, diatoms like these chain species here, as well as these solitaries, are extremely important and abundant, but we also see substantial numbers of solitary flagellates that contribute to biomass and are important in transferring organic matter through the food web. We also capture lots and lots of microzooplankton. Here I'm using just as an example, Foraminifera. These are planktonic amoebae, planktonic or benthic amoebae, which are very important in capturing bacteria, other particles, and small phytoplankton, and transferring them up the food web. This figure at the upper left is an image of a live foraminiferan. They're beautiful organisms. They are amoebae that secrete a shell, and then they exude very long filamentous pseudopodia, which they use in capturing organic matter. Now, the CTD rosette and its 10-liter bottles is very effective in capturing small particles like that, but it's completely ineffective in sampling bigger mobile organisms. For that, we use net systems. This is an image of a mock nest. A mock nest is one of the most cleverly named gizmos we use. The name is an acronym. It stands for the Multiple Opening and Closing Net and Environmental Sampling System, and it's basically a trawl system in which there are multiple nets within a single frame. This particular mock nest I'm showing you is a double mock nest. We often use single mock nest systems in which there's just a single frame with a series of bars suspended in that frame. Between each pairs of bars is the mouth of a rectangular net. On the frame itself is a set of sensor packages. So we tow this net system through the water, and as we do so, we collect physical and some chemical information on the water column, and we sample animals in the mouth of the net, through the mouth of the net that's currently open. We will typically tow this net system diagonally through the water column, periodically tripping nets, that is, closing one and opening the next as we go. This allows us to sample multiple depth horizons in the water column, and then ultimately, usually students will painstakingly sieve and subsample a net tow in order to provide us with samples of animals of different size classes. So the kinds of things that we sample include gelatinous animals. These are some wonderful drawings and watercolors of siphonophores, which are gelatinous animals that are voracious predators and very abundant in the upper water column of most open ocean systems. We also collect lots and lots of copepods. Copepods are crustaceans which are numerically probably the most abundant metazoan, multicellular animal in the oceans, and they form a critical link between the primary producers and the big fish that most people care about. We get a variety of other animals, and the only other one I want to show you are pteropods. Pteropods are flying snails. They are snails which are modified to live in the water column. The line drawings in the middle show in schematic form how these things actually fly. If you can imagine taking a small snail, turning it upside down, and then having it develop big expanded wings that are lateral extensions of the foot, then you have a reasonable image of how a pteropod works. They flap these extensions of their foot, and they are sometimes called sea butterflies because as they flap their feet, they bounce the body up and down in the water column and move themselves very, very effectively. Now, pteropods are particle feeders, suspension feeders. They exude a mucus net, capture particles, and then periodically reel that net in and consume the net and captured particles together. I'm telling you this because you've now seen examples of a carnivorous animal, the siphonophores, animals which may be omnivorous or carnivorous, those copepods, and animals which are primarily herbivorous in the sense that they are particle feeders, capturing particles with a mucus net and consuming them. Pteropods are little known outside of the community of people who really care about and study the ocean, and yet they're a very important link in moving carbon and energy from primary producers up the food web. And interestingly, we know that there is a very important impact of the Deepwater Horizon spill on pteropod populations. So remember, oceanic and other food webs are complicated. Any of the animals I showed you would be embedded in a web, a network similar to the one I'm showing you here, which applies specifically to cod. And it's really hard to tease apart the relationships among them. Thankfully, there are ways to simplify and attack the food web in a way that allows us to discern what the large-scale patterns of energy and material flow are and how organisms are connected to their sources of carbon and how they can pass those sources of carbon through their bodies and onto other organisms. I told you at the start of this that I work at the interface between biology and geochemistry. We use a geochemical tool to understand how food webs, how organisms within marine food webs are connected to their food sources and how they pass organic matter through their bodies. The particular tool we use, in most cases, involves measurements of the isotopic abundance, the isotopic composition of carbon and nitrogen in organic matter. Now, it turns out that both nitrogen and carbon, and today I'm going to focus on carbon, have more than one stable, naturally occurring isotope. In the case of carbon, carbon-12 is the common, most abundant isotope, but carbon-13, which is stable, comprises about 1% of all the carbon on our planet. It turns out that biological and chemical processes discriminate between the isotopes and create interesting and interpretable variability in carbon-13, carbon-12 ratios within ecosystems. Analytically, it's hard to measure the carbon-13, carbon-12 ratio directly with enough precision and accuracy to actually resolve the differences we're interested in. So we use a normalization, and we typically report both carbon and nitrogen isotope abundances using this convention that I'm showing you here. The details aren't important except that you're going to see delta C13 multiple times. The delta C13 of a substance is the isotope ratio, C13 to C12, in that sample divided by that same isotope ratio in a reference compound. Now, because that ratio of ratios is very close to 1, to make this thing numerically simple, we subtract 1 from that ratio of ratios, and then to get the decimal place in a convenient position, we multiply by 1,000. Because the isotopic standard we used for carbon was an inorganic carbonate, virtually all biologically interesting delta C13 values are negative. It doesn't matter. More positive going delta C13s have more C13 in them. More negative going ones have less C13. Because of the standard we use, all of the values will be negative. And I'll show you a variety of these delta C13s. C13 turns out to be a good tracer for carbon sources. This is a classic figure from a study carried out in the Gulf of Mexico and elsewhere. And the point of this is that there's a very, very good correlation between the delta C13 of an animal and the delta C13 of its diet. This applies also to primary producers. Primary producers have a delta C13 that reflects a relative contribution of different sources of carbon and the isotopic composition of those sources of carbon. So here's an example of how we can use C13 to study the movement of carbon into food webs. This was a very nice study that came out in late 2010, very soon after the Deepwater Horizon spill occurred. We're going to start with that upper panel on the left, panel A. To orient you, Mobile Bay is right here, just above that red box. Here is the Birdfoot Delta, and New Orleans is off to the left of our figure. So the study region for this paper is outlined in red there. Panel B shows two very important and interesting pieces of data. The black line shows, in rough terms, the distance of surface oil slicks from that study area. So where the black line is near the horizontal axis, there was oil in or very near that study region. Where it's far away, as off to the right, the oil slicks were very far away. The red line there shows the area covered by oil slicks at different times during this period in 2010. I want to jump now to panel E, the lowest one. Here we have delta C13 of particles collected within that region south of Mobile Bay between June and late August, early September of 2010. Delta C13 ranges in this plot from about -21, -22 to perhaps -27 or -28. Remember, all of these values are negative because of the isotopic reference compound. Typical marine organic matter has a delta C13 of around -18 to -20. The oil coming out of the Deepwater Horizon had a delta C13 of about -27.5. What that means is that if we have typical marine organic matter, we ought to see values that are towards the top of panel E, where we have a very heavy contribution of oil or carbon derived from oil. We ought to be close to that dashed horizontal line. And as you can see, as we go from June through July and early August, there's a very clear trend in which delta C13 is lowest in July and August, and then rebounds and is higher later in that time period. So this is evidence for a contribution of oil or oil-derived carbon to the suspended particles in this region during the latter part of the oil spill. If we turn now to panel D immediately above, this is the delta C13 of zooplankton plotted in the same way, showing delta C13 as a function of time. Now, as you can see, when we get to the latter part of the oil spill, we see very low delta C13s in animals. This is an indication that oil-derived carbon was moving not just into the suspended particles, but also into the food web, that animals, through consumption and metabolism, were capturing some of that oil-derived carbon and transferring it at least part of the way to those charismatic, economically important species that we typically worry about. All right, I'd like to now share with you some of the data from our cruise in August, September of 2010. We're looking at a broader region, so that Graham paper focused on a region immediately around Mobile Bay, right here in the upper part of the chart. I'm going to show you data from all of these stations scattered around the northern Gulf. The Deepwater Horizon site is that red star right in the middle of that cluster of points to the southeast of the Birdfoot Delta. And then we also sampled farther afield. For starters, I've plotted all of the delta C13 measurements we made of suspended particles. And that green line is just a visual reference showing the typical delta C13 of marine organic matter. And when you look at this, I've got depth plotted on the vertical axis, delta C13 on the horizontal axis. As we go down through the water column, you can see a great deal of variability. There's certainly some evidence for low delta C13 particles right up at the surface, which is what we would expect from that pattern that Monty Graham and his colleagues showed. But we also see some rather low delta C13s at depth. A lot of scatter around that typical -20 value. If we look closer at the data, we actually see a very interesting pattern. So in this slide and in the next one, I'm going to separate the data into two sets. First of all, we're going to look at data collected very near the Deepwater Horizon site. So here, from these stations very close to the wellhead, we see that surface low delta C13 signal, as well as a hint of some lower delta C13 values deeper down in the water column, but the distribution is actually fairly symmetric around that -20 value. So near the wellhead, at this point in time, about a month after the major spill ended, we have some evidence for movement of carbon from the well into the suspended particles at the surface and deeper down. Further afield, when we look at these stations that were actually 100 or more nautical miles away from the wellhead, we see a very different pattern. In fact, we see a pretty uniform offset towards lower delta C13 values throughout the water column, with a few exceptions. We see that surface signature still, but now we see very, very clearly a deep signature that is consistent with oil and gas moving into the food web. Now, this is interesting, and in fact, these samples were collected in a region where we know that there was a deep plume of oil and gas that moved to the southwest at a depth of about 1,000 meters. So this is geochemical evidence for the movement of oil and gas carbon into the base of the food web. Now, these are images, these are figures from a manuscript that is currently in preparation by a postdoc in my lab. And the left-hand panel shows the data that I just shared with you. The format's a little bit different, but the white symbols, the open symbols, are from the portion of the cruise well removed from the wellhead. The filled stations, the filled symbols, are near the wellhead. And you can see that isotopic contrast, and in particular, that very clear signature associated with that deepwater zoom at about 1,000 meters. The right-hand panel shows data from samples collected a year later. Very different distribution, and in fact, we no longer see that clear association of low delta C13 values with that deepwater plume. Instead, we see a much greater spread of those low delta C13 values throughout the water column, and this is evidence that the movement of organic carbon into the food web had an impact that lasted not just a few months, but in fact persisted for at least a year, and in fact I'll show you in a few moments, for more than a year in the water column of the deep Gulf of Mexico. Well, what about zooplankton? If we want to understand the food web, we really have to look not just at the particles, we have to look at the animals. And as I told you, we use a moth nest to capture animals. We then separate the animals into different size classes, and we look at the isotopic composition of those animals as a way to understand the movement of carbon from various sources into the food web. Now, before I show you the data, I want to show you a couple of images. First off, in 2010, the samples looked odd. They actually smelled a little odd. So when we process our samples, one of the things we do is we take splits from our nets and we freeze them for later isotopic analysis. What I'm showing you here are the samples from two different nets. The uppermost net sampled from the surface to 24 meters depth. The lower set of bags contain samples from the next lowest net, which sampled between 24 and 44 meters depth. These are different size fractions of animals, formed from animals between 200 and 500 microns up to animals bigger than 2,000 microns in size. And look at the color, particularly of the 500 and 1,000 micron size fractions in that uppermost net. We've never seen that kind of coloration of zooplankton in the Gulf of Mexico except during this cruise. The lower image shows you the typical pattern. Copepods tend to be a pinkish color. That's because of pigments that they obtain from their food. That dark coloration is associated with an oily smell and reflects the assimilation, the aggregation of oil and oil-derived carbon by these animals. The other thing I need to tell you or remind you of is that animals in the ocean carry out a very regular, very well-defined migratory pattern that looks like this. This is actually an echo sounder record. It goes through, it goes from midnight one day to noon the following day and midnight the following night. It's taken in a fjord somewhere up in Norway, and what you can see is the behavior of different layers that reflect sound. These turn out to be a variety of animals, including krill or euphausiids, which form that thick band that you can see down here at about 150 meters depth at midday, as well as three different layers of animals that turn out to be this hydromedusa you can see over at the right. The important point is that this kind of behavior is very, very widely distributed, and this pattern is very consistently found in oceanic water columns. lots of animals come to the surface at night and descend during the daytime. So during the day, they're somewhere deeper in the water column. At night, they're right up here at the surface. Now, I'm going to show you data from a few stations, and we'll start with this one. The panels will be arranged in the same way for the next few slides. In each case, we're going to have two different net tows. These were carried out at roughly the same place, within a nautical mile or so of each other, but at different times. The left-hand panel will show the daytime tow, the right-hand panel, the nighttime tow. Depth is on the vertical axis, delta C13 is on the horizontal axis. So let's start with this daytime tow. This was done at a station about 17 miles away from the Deepwater Horizon wellhead, just about a month after the wellhead was actually closed off. This is a region where there was a lot of surface oil during and immediately after the spill. And in the left-hand panel, I have a plot that shows the delta C13 of different size fractions as a function of depth. And I'd like you to focus on that red line, which shows the smallest size fraction of animals. And you can see that they show the most negative delta C13 of any animals anywhere when we look at the surface sample. Now, low delta C13s reflect incorporation of oil or gas-derived carbon, and what this pattern suggests is that animals that persist at the surface all of the time, so small animals that don't migrate have assimilated more oil and gas-derived carbon than anyone else. Now, if we look at the nighttime tow, and again, focus on that red line, it looks very different than the daytime line. It shows actually rather positive delta C13s right at the surface. There's a decrease in the delta C13 of these animals in mid-depth. So if we compare these data with these data, the nighttime delta C13 of the small animals between 25 and about 200 meters is actually lower at night. So what has to be happening is that animals are migrating vertically, and the small animals that are migrating vertically from this depth horizon between 25 and 200 meters are isotopically heavy, have a higher delta C13 than the ones that remain behind. So that the non-migrating animals have a lower delta C13 than the migrators throughout this upper part of the water column. In other words, animals that are staying put end up assimilating more oil-derived carbon than animals that are migrating. Presumably, this reflects the opportunity for those migrators to get at other sources of carbon and to dilute out that oil and gas-derived carbon that the small non-migrators are assimilating. Here we are at point 19 miles southwest of the wellhead, and we actually see a rather different pattern. Now, this region saw some surface oil, but it's also an area where we had a very clear impact of that deep water plume that moved away towards the southwest from the wellhead. Again, focusing on the smallest animals, the pattern is actually rather different than we saw to the northeast of the wellhead. As we go from daytime to nighttime, surface animals actually show a lower delta C13 in the nighttime than they do during the daytime. In other words, the migrators, the ones that are coming to the surface at night and going downward in the water column during the day, have a lower delta C13. This suggests that the main point where carbon from oil and gas was incorporated was not at the surface, but was deeper in the water column, and the distribution of these values vertically suggests that it was somewhere within the upper few hundred meters of the water column. if we go yet farther to the southeast, we actually see relatively little change in the upper water column between the daytime and the nighttime tows. The place where we actually see an interesting shift is down here at the very deepest area that we sampled. And it's not as pronounced in the small animals as it is in some of the larger migrators. Now, the only way that we can, or the most likely scenario for acquiring this kind of rather low delta C13 deep in the water column is for that to reflect animals that are migrating from the deeper and coming up to this horizon about 300 to 400 meters depth. In other words, we are seeing the impact of even deeper carbon that we sampled on the deep pelagic food web out in this region. Now, this station turns out to be in a really interesting location. It's close to a major seep, and it's an area that we've sampled repeatedly. If we look at what happened a couple of years later, so this is in 2012, at the very same location as this station, but here we're able to sample much deeper in the water column. So rather than looking just at the upper 400 meters of the water column, now we're looking at the upper 900 to 1,000 meters. Unfortunately, we only have smallest size fractions for the daytime tow. We haven't finished processing all of the other animals. We have all of the size fractions for the nighttime tow. The important point, the one I want to make, is that the changes between daytime and nighttime are rather subtle and nothing like the shifts we've seen at other times at other stations immediately after the Deepwater Horizon spill. This suggests that those large contrasts we were seeing immediately after the spill were in fact a reflection of that very, very large infusion of oil and gas and not characteristic of the Northern Gulf ecosystem in general. So if we look at the data I've shared with you, the major points I want you to take away are that stable isotopes can and do record the movement of oil and gas carbon into the food web, and in fact they provide us with our strongest and most robust tool for understanding how oil and gas can affect the pelagic food web. And just to summarize, we saw low delta C13 in particles immediately after the spill, and this reflected both the direct contribution of carbon, of oil to the particle field, and the movement of some of that carbon into the particles through heterotrophic activity. That low delta C13 signature persisted for at least 10 months and in fact appeared to spread through the water column over that 10-month period. We know that oil and gas-derived carbon made its way into the food web, and we have some isotopic indications that this food web impact persisted for at least a year and a half, close to 2 years after the spill. I'd like now to just run quickly through some of the broader impacts and then show you some of our very recent research that tells us something more about how rapidly the system can respond to spills. Now, I told you early on, I mentioned early on, that we know that the food web impacts cascade not just through the food web up to big organisms, can cascade downward through the water column and all the way to the sea floor. We've been studying these vertical connections in a variety of ways, and one of the tools we use is a sediment trap. The upper left figure shows a large time series trap that's being deployed. That's a trap we deployed in 2010, very near the wellhead. We've retrieved it and turned it around repeatedly, and in fact now have one of the best records of the sinking of organic matter in the Northern Gulf that anybody has. And when I say we, I'm now meaning the EcoGig Consortium, This isn't my personal work, I'm involved in it, but in fact, this part of the project is led by other members of the consortium. We also use floating sediment traps, like the one shown at the lower right, which we can deploy for a period of a few days, and these sample the sinking organic matter at about 150 meters depth and allow us to really tie what's happening in the upper water column to what's sinking into the deep sea. We also use multi-core, a device that lets us collect samples from the sea floor. These are two of my colleagues, Mandy Joy and her former postdoc, Melissa Cristalina. Immediately after we recovered the multi-core from very near the Deepwater Horizon wellhead, the multi-core in this case is full of mud, and though it's hard to see right now, there's actually a very thick and very well-defined layer on the sea floor and in the upper part of those cores that's that has a higher content. So here's the multi-core on deck, and there's Melissa taking some photos, and you can just make out that thick flocculent layer that has a different color than the mud further down in the core. And in fact, if we zoom in, that thick flocculent layer contains a lot of biomaterial, a lot of mucus, and interestingly, it contained a lot of these shells. Remember I told you that pteropods are important grazers and that they are an important link in the food web. Well, these are pteropod shells. Here you can see the little spines on the bottom of that shell. That particular shell is right at the surface, and this one's deeper down. There was clearly a very high mortality of pteropods in the water column, and we know very little about what the impact of that mortality was on the movement of organic matter through the food web to higher trophic levels. And yet it was clearly a mass mortality. For us to see pteropods in every one of our core tubes, virtually everywhere we sampled it in the Northern Gulf, had to mean that there was a tremendous and very unusual mortality event for those organisms. So this core top layer was everywhere within some 20 to 30 nautical miles of the wellhead. We know that it was a very recent event. There were lots of dead organisms, including macrofauna, worms, and crustaceans, and that organic layer contained lots of oily characteristics. Organoleptic is a word I learned at a NOAA meeting. It's a fancy way to say that the stuff smelled and or tasted like oil. I didn't taste it, but it certainly smelled like oil. Microscopically, we could see globules that resembled degraded oil, and we know from experimental work that this flocculent layer not only killed macrofauna, but it had a pretty strong impact on microbial processes down in the water column. All right, the last thing I'd like to share with you is some work that we carried out in a rapid response mode in 2013. In late July, we were actually ending one of our cruises when the captain came into the lab and told me that he was hearing a bunch of Coast Guard radio traffic about a rig that had exploded. And in fact, this rig shown at the upper left had just exploded on 23 July. They lost control of one of their wells. It was a gas production rig. They lost control of one of their wells. The rig caught fire. They abandoned it. Thankfully, 40-some people had to go into the water in lifeboats, but they were all rescued. No, there were no fatalities. We didn't have time to take our research vessel back to the wellhead, which was in yellow here. At the time, we were just south of the Bird-Foot Delta heading back to Gulfport. But we were able to mobilize a rapid response cruise using another vessel out of Hercules, Louisiana, and we were able to sample in the vicinity of that wellhead between 27 and 30 July. So just a few days after the rig exploded and a few days after the well self-sealed. These are data from a manuscript currently in review. It was written by one of my students, Sarah Weber. I'm going to focus on just a couple of the panels here. The upper left panel, panel A, shows oxygen concentrations at a variety of locations within 6 or 7 nautical miles of the wellhead. Now, two things to note. There's a huge amount of variability on that spatial scale. So the wellhead was injecting oil and gas, mostly gas, into a water column that's very dynamic, and yet several days after that injection ceased, we see a lot of lateral variability, a lot of spatial variability. And in particular, we can see evidence of significant oxygen reduction in several layers of the water column, not uniformly, but in locations that were close to that wellhead. Oxygen drawdown is associated with high metabolic activity, and in fact, we have other evidence that suggests that this is one of the signatures of metabolism consumption of the gas that was released. And again, note how different profiles are that were separated by just a few nautical miles. The impact of that gas injection was very localized, and yet it was very dramatic for waters that received that injection. Here are some profiles we have from very near that wellhead. Those light gray stripes correspond to the ones in that oxygen plot where we saw significant oxygen depletion. Remember, -20 or so is characteristic, is a delta C13 that's typical of marine organic matter. -22, -23 is characteristic of organic matter that has a contribution, a measurable contribution of oil or gas carbon. And in fact, that contribution is associated with the regions of the water column where we saw a significant drawdown of oxygen. We've done a variety of other measurements and calculations, and we can associate this oxygen drawdown with carbon consumption and incorporation into the food web. And in fact, our data imply that anywhere from a few percent to perhaps 20 to 30% of the carbon that was present in the organic matter in the water column was derived from the gas that was released over that 2-day span. So there was a very rapid, very pronounced impact on the base of the food web. What we don't know, because we weren't able to collect animals, is how that propagated into the food web. But we know that the base of the food web at least can respond on a time scale of days. So to wrap up, when we study the Gulf of Mexico or really any oil spill, we have challenges. We're dealing with a complex system with multiple interconnections and with some real challenges in sampling the ecosystem and in sharing our science, sharing our work with the general public. We have an additional challenge, and that is that a lot of the organisms that are important just aren't that familiar, and in some cases aren't that interesting to the general public. So we always have a hurdle in trying to share our findings on critical organisms with the general public. When we go broader afield and try to share our work with journalists and policymakers, we have another level of challenge in that everybody likes simple answers and very clear, definitive statements, but in fact, it's hard to make simple answers and definitive statements about complex systems. And I always like to emphasize to people in science and out that science is a fundamentally iterative process where we always are refining our hypotheses based on the samples and data we have, and that we are always constrained in what we can say about anything by the available data, by the available state of knowledge. So I will end there. I'll thank you for listening. I haven't explicitly acknowledged all of the people that contributed, but again, Mandy Joy is a colleague that has been involved in many of our efforts at sea, and in fact is the leader of the EcoGig consortium. Natalie Lowell-Field and Julie Gonzalez were members of my lab who took part in the field work. Jason Landrum is a former student who took part in much of the field work. Sarah Weber is a current student who led much of the particle sampling that I showed you. And Aliza Bracco is a physical oceanographer, and Ajit Subramaniam is a satellite oceanographer. They're both members of the EcoGig consortium, and both have been very important in helping us refine our understanding of the broader context for our measurements. I will stop there and would be happy to take any questions that you might have. And thanks for listening.
Well, thank you very much for that presentation. So as I said earlier, everyone, we have about 30 minutes now set aside for question and answer. So if you just use the chat box in WebEx, feel free to type in all your questions, and I will relay them to Dr. Montoya so he can answer them for you. And these can be questions on his presentation, maybe things that you need a little more help with in preparing your students for this topic of the science of oil in the oceans, or even if you have questions on how he got to where he is today through his his career and his school choices. So we'll just wait a minute for people to type in their questions.
I have the chat box open, so I can see anything that you type in, and I will tell you that I'm used to people finding the work we do a little cryptic and a little times hard to get excited about. So don't be shy if you have questions about stuff that may seem trivial or may feel unrelated. I'm happy to talk about any aspects of our work or more broadly about biological oceanography and the ways that we attack problems in food webs and pelagic ecosystems. And I have to say, I teach introductory biology, and I often have to walk around picking on people to get them to open up. But usually they will ask questions in the end.
Well, hopefully by providing that, emphasis on anything related to what you do and, biological oceanography, I hope we'll have a few questions come in. So, again, we'll just give everyone a minute or so to get their questions typed in. Okay, so the first question came in and they're asking, could you explain again if there's any difference in the D13C in oil from natural seeps versus Deepwater Horizon oil?
It's an excellent question. The -27, -28 that I showed you is typical for oil from the Deepwater Horizon site. There is variation in the Gulf, and that variation reflects the ultimate sources of the oil. But there's not a huge amount of variation. Oil in general is depleted in C13 relative to marine organic matter. It can vary from -27 or 28 to -30 or thereabouts. Now, the reason that oil is isotopically depleted relative to marine organic matter is that oil carbon is ultimately derived from, in large part, from terrestrial organic matter. And terrestrial organic matter, because of the photosynthetic pathways that lead to carbon fixation, is typically isotopically depleted relative to marine organic matter. The ways in which organic matter are formed are different, and we end up with different isotopic signatures. So there is some variability in the delta C13 of oil from different sources, but it's relatively small compared to the difference between oil delta C13 and typical marine organic matter delta C13. To complicate matters, gas released from the seafloor has a very different isotopic composition. So remember that average marine organic matter is at about -20. Deepwater Horizon oil was at about -27 or -28. Methane released from the seafloor can have a delta C13 as low as -60 or -80. And that's a reflection of the pathways that generate the methane. Most of the methane that was released from the Deepwater Horizon was at the higher end of that range, about -30 to -40. But there are places in the Gulf where we can find methane being released through natural seepage that has a delta C13 that's much, much lower. So depending on where you are, and especially if you're in the immediate vicinity of a seep, you might see much lower delta C13s. But in the way that we surveyed broadly, we typically don't expect to see the localized impact of seeps. And for the 2010 cruise, we're pretty confident in attributing the delta C13 signatures we saw to the impact of the Deepwater Horizon.
Okay, someone asked if you have any speculation as to how your impact or findings might differ in the Arctic waters.
Another excellent question. Now, if we were working in the Arctic, I would expect a difference in the way that oil was partitioned between the sea surface and the deeper water column. And the reason I say that is that much of the movement of oil downward in the water column was biologically driven. There are microbial and phytoplankton processes that help to aggregate, to trap oil, aggregate it, and package it in a way that it can then sink rapidly towards the sea floor. In the Arctic, because of the lower temperatures, I'd expect that to happen much less rapidly. So I would expect more of the oil to end up at the surface. I would expect less to go down to the sea floor. I don't have as good a feel, or it's harder for me to predict how any subsurface plumes, subsurface injections might act. My instinct, my inclination would be to say that subsurface injection at high pressure, in the same way that occurred at the Deepwater Horizon, should produce neutrally buoyant clouds of small oil droplets, which would then move laterally, and I would expect a slower metabolic response because of the lower temperatures. Now, one of the things that we know happen in the Gulf is that populations of microbes capable of consuming oil and gas became active, metabolically active very rapidly. Their populations increased very rapidly, and they played a key role in consuming and drawing down the oil and gas released into the environment. Whether there are similar populations in the Arctic, we don't know. It's likely that there are at least some microbes that can consume oil and gas out there, but how active and how abundant they are, we don't know. But my guess would be that because of less environmental exposure, natural environmental exposure to oil and gas, that they would be less abundant, less active, less primed to take off in the way that they did in the Gulf. So that was a very long-winded way to say that my expectation would be that in the Arctic, the biological response would be slower. There would be a slower packaging and transport of oil-derived carbon from the sea surface to the sea floor, and that the overall biological response would play out on a longer time scale. So rather than the week-to-month time scale of significant impact in terms of movement of oil and gas into the food web, I would expect it to play out over a time scale of months to perhaps years.
Okay. I think this question may have been in relation to something you had noted earlier, but someone was asking, how long might it take for the environment to correct itself?
Another excellent question, and it's one that's really hard to answer in a general way. My colleagues in the EcoGig consortium are carrying out a whole suite of time series measurements. One of them involves looking at deepwater corals, and they have repeatedly photographed and measured coral colonies in the northern Gulf. And one thing I can share with you is that immediately after the spill, they were able to document shifts in, let me say it differently. They were able to document direct damage to coral colonies that had been snowed upon, that had oil, marine snow with oil content landing on them. There was a flux event in which significant quantities of oil-derived and oil-affected organic matter rained down to the seafloor, and they were able to document an immediate effect on these deepwater corals. They've been back repeatedly to survey these corals, and qualitatively they could see morbidity of these coral colonies, necrosis, and some dieback of the colonies over the first year to year and a half. They've seen some recovery of the coral colonies since then, and by recovery I mean some regrowth in the areas that have been affected by the oil. So this is anecdotal on my part. They've quantified this and actually have some very good measures of the rate of recovery, but it's a partial recovery in the sense that the regrowth doesn't match the loss that occurred in the early stages of this impact. In other areas, we know that the sedimentation event had a clear impact on microbial communities within the seafloor. Recovery appeared to take, there was some recovery of microbial metabolism after a few months, but there is still a very large alteration of microbial metabolism in the seafloor in the areas where there was significant deposition of organic matter. Our data up in the water column suggests that there was, I don't want to say a recovery, but that the input of carbon from the Deepwater Horizon was detectable for the first year and a half to perhaps 2 years, but then was diluted out within the water column ecosystem by more recent production and assimilation of organic matter. That doesn't mean that the impact is gone. It just means that the direct input of carbon has now been diluted out by production and assimilation since the spill ended. When we look at other trophic levels, it becomes even trickier, even harder to predict what the time scale, what the trajectory of recovery is and will be. As an example, we know that the shrimp fishery suffered in the immediate aftermath of the spill, but we also know that the shrimp fishery appears to have undergone some significant recovery since then. Same for the oysters. For some of the big pelagic fishes, there's a real concern in that they have life cycles that involve multiple years from egg till the larvae are mature enough to recruit into the fishery. For example, tuna need 3 to 5 years from the egg until they're big enough to actually be caught and part of the fishery. So that means that for some of these big pelagic species, we'll only now be seeing the year classes that were actually affected directly by the spill entering the fishery. So we don't yet know what the impact is on these these higher trophic levels. And it may well be that the system has recovered, or it may be that we're only on the verge of starting to see the direct impact of the spill on these big, important species.
Okay, thank you. Some of the students, I think from Cypress Woods High School, were asking, what was the rationale for the locations chosen for sampling far from the well? To them, it seemed mostly random. They were talking about US slide that had sample sites shown.
Okay, in 2010, and I actually see the comment and the request to move back to the sample slide. Let me see if I can get there as I talk. The rationale was that we wanted to sample near the wellhead, but we also wanted to survey and gain some understanding of areas farther afield. Oops. I guess this is the slide that we want. I actually was the one who laid out this cruise track, and the way that we did it was we started in Gulfport, and we wanted to begin by surveying in areas where we would be least likely to contaminate or damage our equipment. So we started with this line right here. This was our first sampling site, and we worked from east to west, the idea being to run a transect some 80 to 100 nautical miles south of the Deepwater Horizon site. At the time, we had every expectation that we were sampling waters completely unaffected by the Deepwater Horizon spill. If you look at satellite imagery, there was lots of evidence for surface oil up in this region to the north and northeast of the wellhead. There were well-defined oil slicks that moved to the east and, to the west and southwest of the wellhead. There was no substantial movement of surface oil to the southeast. So these stations were ones that we envisioned as sort of control stations, areas where we had little likelihood of an oil impact. In fact, we later found that there was substantial evidence for a deeper impact of the spill, not at the surface, but for movement of oil and gas deep down in the water column to the southeast, not just to the southwest. So we began by running this line. This cluster down here is a set of stations where we were looking for and surveying that southwest plume. So a deep plume left the Deepwater Horizon wellhead and moved to the southwest at a depth of about 1,000 meters. And these stations down here to the southwest were ones where we were sampling and capturing the plume. We then went into port, we swapped out some people, we came back out, and we repeated our survey around the wellhead, but on a smaller scale. We started by running a box, a rectangle around the wellhead, did some focused sampling in areas where we knew that there was substantial surface oil, and then got in as close as we could to the wellhead. So the basic sampling strategy was to first survey far afield, try to get at waters that were unaffected by the well, by the spill, sample the southwest plume, and then on the second leg, get in as close as we could to the wellhead itself.
Okay, I think we have two more questions. One's asking, what conclusions can you make based on your research about the effects of the Deepwater Horizon spill on productivity in the Gulf? And they also asked, have any satellite studies been done?
Okay, productivity is actually a hard question to answer. And let me start by saying that we measured productivity and we measured chlorophyll concentrations on our cruises. We also have access to satellite imagery, satellite data that allow us to quantify the amount of chlorophyll at the sea surface. Now, one of the things that has become apparent is that immediately following the spill, there was actually elevated chlorophyll in the northern Gulf. Now, it's hard to relate that directly to the spill in that lots of things were going on. There was a big release of water through the Mississippi. This was in part an attempt to try to force the spill, the surface slopes, further offshore. So there was a lot of fresh water coming in. Fresh water brings nutrients. It brings buoyancy so that it in itself can promote higher chlorophyll concentrations and higher productivity in the northern Gulf. So there is some satellite evidence. We have evidence from our cruises that phytoplankton biomass, phytoplankton productivity may well have been elevated immediately after the spill. Now, I'll share with you some other work that's been ongoing within the EcoGrid Consortium, and it's work carried out primarily by our colleagues at Columbia University, led by Ajit Subramanian, whose photo I showed you a moment ago. And using a variety of sensors, they actually are developing some pretty compelling evidence that injections of oil and gas directly lead to elevated chlorophyll concentrations in areas around seeps. Now, this relates to our observations immediately after the spill, but on a smaller scale and with much, much smaller exposures to oil and gas. The mechanism isn't entirely clear, but this is a very exciting finding. The finding has been written up. It's out for review right now, but it's somewhat surprising, very novel finding. We are unsure of the mechanism, but it appears that differential impacts of oil exposure on phytoplankton and grazers is a very important aspect of this. In other words, it may be that grazers are more sensitive to oil exposure than phytoplankton, and that one of the consequences of injecting oil into the environment is that phytoplankton may experience at least a partial release from grazing pressure. Again, this is a work in progress, but the EcoGrid Consortium now has some very exciting evidence that oil releases counterintuitively might actually lead to some enhancement in productivity. Now, how that translates into secondary production, production of animal biomass, and ultimately production of fisheries is a very, very difficult question to address, and we really don't know the answer to that part of it yet.
Okay, I guess the last question was actually just a repeat of the one before, but specifically asking if any productivity studies using satellites with chlorophyll sensors have been carried out.
Yes, and I have to make one distinction, and that is that satellite imagery, remote sensing can give us a measure of biomass of standing stock, so it can tell us what the phytoplankton biomass is. It doesn't directly give us a measure of productivity. In many parts of the ocean, there is a very strong correlation between standing stock of phytoplankton, between chlorophyll concentration and productivity, but that's a correlation. It doesn't always hold. And in fact, we can have changes in productivity that don't show up as changes in chlorophyll concentration. In other words, production rates, the rate of photosynthetic activity, can go up without changing biomass if the grazers are capable of keeping up. So yes, there have been lots of studies. EcoGrid scientists are very actively involved in using satellite imagery to understand what the impact of oil and gas releases are on chlorophyll. It's a little trickier to get at productivity, but by going out in ships and complementing our satellite data with direct measurements of rates, we're beginning to get at that link between oil and gas exposure, oil and gas release, and productivity in the marine environment.
Okay, that was the last question. Was it typed in? Does anyone else? Oh, okay, one came in. What is the average number of new species of animals or plants that you find once a month?
I have to confess, I'm trained as a biologist, but I work as a geochemist or biogeochemist. And I don't actually personally discover new species. I may have helped in the discovery of a couple of new coral species through some of our sampling activities, but I'm not a specialist in those, and it's my colleagues who actually are able then to take the samples and characterize them, describe them if they're new, and name them. So even though I'm a biologist by training, I rarely work at the species level, and most of my work involves looking at geochemically interesting and relevant parts of the ecosystem, whether it's particles which contain lots and lots of interesting and beautiful phytoplankton, or zooplankton toads from our nets, which contain lots of beautiful and interesting zooplankton, but I'm not the one, sadly, who can or is competent to describe or do the sort of species level work that we often associate with biologists. So in my case, the answer is 0 and never.
Okay, that was the last question that came in. So, oh, nope, one more, sorry. At what depth does the light stop getting through the water?
It depends where you are, and the depth of penetration of light depends on what's in the water column. Biologically, the depth at which light is diminished by 99% relative to what hits the surface is very important. It turns out that for mid-latitude ecosystems, roughly 1% of average surface irradiance is what it takes phytoplankton to just break even. If they have more light than that, then they can actually grow. If they have less than about 1% of average surface irradiance, they can't photosynthesize enough to balance their respiration. So we use the 1% light level, the depth at which light decreases to 1% of its surface value, as a rough index to the part of the water column where photosynthesis is a viable strategy for making organic matter. Now, in the Gulf of Mexico, there's enormous variability in the depth of the 1% light level. If you're near shore in the Mississippi plume, the 1% light level might be a few centimeters below the surface. The data I showed you for the Hercules ridge study, in that area, the 1% light level was about 1 to 2 meters deep. So not very deep at all, about 3 to 6 feet down. In open waters of the Gulf of Mexico, so if we get out here beyond the shelf break, then we're actually in waters that look very much like the open Atlantic or the open Pacific. And it's not uncommon to have a 1% light level that is 50, 60, sometimes as much as 80 or 90 meters down in the water column. These are waters that are deep, deep blue, have relatively low chlorophyll and particle concentrations, and really do resemble. They're very much like the open Central Ocean in the Pacific or the Atlantic. So it depends where you are, and in the near shore regions, water may penetrate only a few centimeters into the water column. Well offshore, it can penetrate many tens of meters.
Okay, well, thank you very much for answering all those questions. We've run up to the end of our 30-minute question and answer session. So on behalf of everyone who participated today, I'd like to say thank you, Dr. Montoya, for your presentation. Just so everyone knows, we will have the recording of this webinar posted on our website shortly. All you need to do is go to www.nosb.org, and on our front page we have a link to the webinar series, and we have the recordings for all 3 of our presentations. This was our 3rd and final part of the series, so please go, if you haven't seen the past 2, please go to our website and watch those. We will also be posting these recordings onto our NOSB YouTube station shortly, so you will also be able to access them there. Again, I'd like to thank everyone for participating today, and again, thank you very much, Dr. Montoya.
Thank you. My pleasure. I had a good time.
Great. That's what we love to hear. All right. Thanks, everyone.
Okay. Thank you, Melissa.
Thank you so much.
Anything else I should do?
Well, I'll send you an email to see if you have any ability to write some questions based on today's webinar.
Sure.
That we can incorporate into the competitions. That would be great. But I'll send you an email with that information.
Okay. Happy to do it.
Yeah, and that's about it. And I will also send you the link to where we have this recording posted so that you can watch it yourself or share it with others.
Sounds good. All right. Well, thanks again.